It's pretty wild when you think about it, how something from way, way back in the past can still affect us today.
Oh, absolutely.
And that's exactly what we're diving into, this incredible link between, ancient DNA from people thousands of years ago and how we actually understand and even treat a modern disease like HIV.
It sounds almost like science fiction.
Doesn't it? Some people, believe it or not, have this genetic quirk, this tiny change that gives them real protection, maybe even immunity against HIV. It seems futuristic, but its story starts deep, deep in the past.
Yeah, and what's so cool here is it's not just some random fluke. We're actually tracing the evolutionary history of a very specific genetic change. It's a deletion, a missing bit of DNA
Yeah.
called CCR5 delta 32.
CCR5 delta 32.
And recent research has managed to piece together its whole story, and it's a pretty unexpected one.
Totally. And we've got some fantastic research guiding us today. There's a really detailed paper in the journal Cell from this year, 2025, and also a great summary piece on ScienceX from May 8th, 2025, breaking it all down. What really struck me was how they did it. They didn't just look at modern DNA.
No, they went way back.
They used these super advanced techniques on ancient DNA and then combine that with AI methods to sift through all the data. We're talking genomes from today and genomes from millennia ago.
It really highlights how far things have come scientifically. I mean, pulling genetic data from ancient remains and connecting that to modern disease resistance is just wow.
Yeah, it's a powerful example of connecting different fields.
Exactly.
Okay, so let's get into it. The CCR5 delta 32 deletion, what exactly is it? What's it doing in our bodies?
Right. So think of the CCR5 gene as the instruction manual for building a specific protein. It's a receptor, kind of like a docking station that sits on the surface of some of our immune cells.
Okay, a docking station. I like that.
Now, the CCR5 delta 32 deletion is a specific typo, you could say, in that instruction manual. A small piece is missing just 32 DNA base pairs. Tiny, but it has a big effect. Because it's missing, it causes the protein building process to stop too early.
So the docking station doesn't get fully built.
Exactly. It's incomplete.
Yeah.
Or it might not even make it to the cell surface properly.
Right.
And here's the crucial part for HIV-1.
Right.
That virus, HIV-1, actually needs that CCR5 docking station to grab onto the immune cell and get inside.
So it's like the key fitting the lock.
Precisely. So if someone inherits 2 copies of this deletion, we call that being homozygous, their cells basically lack these functional docking stations.
Okay.
And that means HIV-1 has a really hard time getting in. It provides significant resistance to infection.
That is genuinely amazing. A tiny genetic accident from who knows when, acting like a shield against a modern virus. But I remember reading it's not all good news, is it?
That's a really important point. It's a classic case of what scientists call pleiotropy.
Pleiotropy.
Which just means one gene or one genetic change can have multiple different effects on the body. So while CCR5 delta 32 protects against HIV-1, it's also been linked to how we respond to other viruses, like SARS-CoV-2.
The COVID virus.
Yeah, and it's also associated with various immune conditions, neurological stuff, even some cancers. So it's definitely a trade-off. An advantage in one context might be a disadvantage in another.
Okay, so it's complicated. Now, the research also mentioned these things called CCR5 haplotypes. What are those? How do they fit in?
Right, so a haplotype is basically a set of genetic variations located close together on a chromosome that tend to be inherited as a block, as a group.
Like traveling companions.
Kind of, yeah. So the researchers looked closely at the CCR5 gene region in European populations using data from the 1000 Genomes Project. And they found 3 main groups, 3 main haplotypes, which they called A, B, and C.
Okay, A, B, and C. And where does our deletion, the CCR5 delta 32, sit?
It's found specifically on haplotype A. But here's the interesting thing. Haplotype A isn't just the deletion. It's that deletion plus a whole bunch of other genetic variants nearby, up to 107 of them, that are often inherited together with it. That's called linkage disequilibrium.
Linkage disequilibrium. So they're linked, they travel together.
Exactly. They're so close physically on the DNA that they rarely get split up during recombination. So in the CEU population they studied, that's folks with Northern and Western European ancestry. Haplotype A has 86 specific tag variants, and among those are 2 specific SMP, single-letter changes, that in this group are always found with the deletion, like perfect correlation.
Always together. So haplotype A is the deletion plus its usual crowd of genetic friends. What about B and C then?
Well, haplotype B has most of those same friends, 84 of the 86 tag variants from A in the CEU group, but Crucially, it doesn't have the CCR5 delta 32 deletion itself, and it doesn't have those two SNPs that are perfectly linked to it there. And haplotype C is very similar to B, sharing 82 of those same tag SNPs. The key takeaway here, maybe, is that the deletion likely arose on an existing haplotype background, probably haplotype B. It wasn't created from scratch.
Gotcha. Like adding a spoiler to an existing car model, kind of.
Yeah, something like that.
So how did they figure out where and when this whole thing started, this deletion appearing on haplotype A?
This is where it's really clever. They developed a new probability model, specifically designed to work with haplotypes in ancient DNA. They called it HAPI.
API, okay.
Think of it like a sophisticated genetic detective. It looks at subtle patterns in the DNA to trace relationships and origins back through time. They used API to scan 934 ancient genomes, mostly low-coverage ones, which are harder to work with.
Wow, nearly 1,000 ancient individuals.
Yeah, and by looking for the specific signature of CCR5 delta 32 within its haplotype context, they traced its origins back to the Western Eurasian steppe region.
The steppe again. Okay. And how far back are we talking?
The evidence points to at least 6,700 years ago. The ScienceX report mentions maybe even further back, possibly 9,000 years ago, based on another analysis method they used called CLUE. So we're talking late Neolithic, early Bronze Age times.
That is seriously ancient history. Did they find actual ancient people with this deletion?
They did. The oldest confirmed sample carrying the CCR5 delta 32 allele was from a place called Protoka in Russia, dated to around 5,800 years ago.
Wow.
And they also found it in a really well-preserved Yamnaya individual, the steppe pastoralist from Karagash in Kazakhstan, around 4,900 years old. Plus, they found it popping up in Denmark over 4,000 years ago.
So it wasn't just in one place. It was starting to spread out even back then.
Exactly.
And didn't the news report say it spread incredibly quickly? That sounds like, well, it must have been really useful to have back then, right?
That's exactly what the data suggests.
Yeah.
There's strong evidence for positive selection. Basically, something in the environment was favoring people who carried this haplotype A with the deletion.
Positive selection, making it more common.
Right. This seems to have happened mainly between about 8,000 and 2,000 years ago in Western Eurasia. They used statistical models like CLUES to reconstruct how the frequency of the allele changed over time, and the signal for selection was significant.
So the allele frequency went up faster than you'd expect by chance.
Much faster. And interestingly, when they broke it down by ancestry, the selection seems to have started earliest in populations related to Eastern hunter-gatherers, EHG, and Caucasus hunter-gatherers, CHG.
Okay, so hunter-gatherer groups initially.
It seems like it. And just to check, they looked at 7 other deletions with similar characteristics, and none of those showed this strong signal of selection. So it seems specific to CCR5 delta 32.
Right. So the big question then is why? What was giving people with this deletion an edge thousands of years ago?
Yeah, that's the million-dollar question. The leading hypothesis is that maybe, just maybe, the deletion helped by sort of dampening down the immune system response in certain situations.
Dampening it. Isn't a strong immune system always good?
Usually, yes. But think about the context. This is during the time humans were transitioning to agriculture, living closer together, higher population densities, probably encountering new diseases.
Ah, okay. More potential for epidemics.
Exactly. And sometimes an immune system that goes absolutely haywire in response to a new pathogen can actually cause more damage than the pathogen itself. Like a cytokine storm.
Right, I've heard of that. So maybe a slightly less aggressive immune response was actually better for surviving some of those ancient bugs.
That's the current thinking.
Yeah.
It might have prevented these really damaging overreactions. And the researchers argue that this early selection pressure during the Neolithic and Bronze Age is likely why the deletion is relatively common in Europe today.
Not the Black Death. I thought people used to link it to the plague.
That was a popular theory, but this analysis suggests otherwise. They didn't see a big jump in the allele's frequency specifically during the medieval plague period. The rise seems to have happened much earlier.
Okay, that makes sense. So it originates in the Western steppe, gives some kind of immune advantage against early farming area diseases, and then spreads out.
Yep, the inference is a pretty rapid spread westwards from that steppe origin point.
And how did it get elsewhere, like the Americas?
Well, its presence in Latin America today seems pretty clearly linked to gene flow from European populations after Columbus during the colonial period.
Right, post-Columbian mixing.
Exactly. And just as a side note for geneticists, those two SNPs that are perfectly linked to the deletion in Europeans, RS1133418449 and RS1130100081, They turn out to be really reliable markers for tracking European ancestry in genetic studies.
Like a little genetic flag. Useful stuff. Now, you mentioned something about being careful. There was another genetic marker people used to use as a stand-in for this deletion.
Yes, exactly. There's a variant called RS738333033. For a while, some studies used it as a proxy, thinking if you had this SMP, you probably had the deletion.
But that's wrong.
Well, it's unreliable. This new research really clarifies that RS738333033 is actually found on the other haplotypes too, like B and C, where the deletion is absent. This is especially true in African populations, where the CCR5 delta 32 deletion itself is virtually non-existent, but this proxy SNP can still be present.
Ah, okay. So using that proxy would really mess up estimates of how common the actual deletion is and where it came from.
Absolutely. It could lead to quite misleading conclusions. You really need to look for the deletion itself or those tightly linked SNPs on haplotype A.
That's a super important clarification for researchers. Okay, so let's bring it back to today, to modern medicine. How does knowing all this ancient history actually help with HIV treatment now?
Well, fundamentally, the reason we even have drugs that target the CCR5 receptor for HIV treatment is because scientists discovered this natural resistance conferred by the delta 32 deletion in the first place. That was the clue.
Right, it showed CCR5 was a valid target.
Exactly. But this new research adds a crucial layer. It really emphasizes that when we design therapies aiming at CCR5, we shouldn't just think about the deletion itself in isolation. We need to consider the whole haplotype, all those other linked variations on haplotype A.
Why? What difference do they make?
Because those linked variants might have their own biological effects, maybe subtle ones, but they could influence how well a CCR5 blocking drug works, or potentially contribute to side effects or interact with other pathways. It's the whole genetic package that matters.
So it's not just about mimicking the deletion, but understanding the entire genetic context it evolved in.
Precisely. Nature selected for the whole haplotype, not just the deletion bit. We need to appreciate that complexity.
Okay, this has been fascinating. Let's try and sum up this deep dive. What are the absolute key takeaways for you?
I think there are three main things. First, the origin. CCR5 delta 22 popped up in the Western Eurasian steppe at least 6,700 years ago, maybe earlier.
Okay, step origin, way back.
Second, the selection. It was strongly favored, likely by ancient pathogens, during the late Neolithic and Bronze Age in Western Eurasia, starting among hunter-gatherer-related groups.
Strong selection pressure back then. Got it.
And third, the modern relevance. Understanding the entire haplotype, not just the deletion, is really important for developing therapies targeting CCR5 because of all those linked genes.
The whole package deal matters. Yeah. It really is just incredible, isn't it, that a genetic tweak from thousands of years ago, something that probably helped our ancestors deal with some long-forgotten disease, is now central to tackling HIV.
It absolutely is. It connects our deep past to our present health in such a direct way.
Makes you wonder, doesn't it?
It really does. And maybe here's a thought for you, listening, to mull over. How many other genetic traits or predispositions that we see today might actually be echoes of the past? Relics of adaptations to environments or diseases that just don't exist anymore.
Whoa. Like hidden genetic stories all around us.
Exactly. That ongoing conversation between our genes and our history, much of which is still invisible to us.
That is a fantastic thought to end on. And if you are fascinated and want to get into the nitty-gritty, you can look up the full study in Cell or check out that Science X report. It really shows how the past isn't even past sometimes.